We usually think of cancer as just chaos, pure random biological chaos. But what if I told you that hidden inside all that randomness, there might be a simple, predictable, even mathematical order? Today, we're diving into a single scientific paper that proposes that absolutely radical idea.
So, let's just start with the big question right up front. What if the messy, destructive process of genetic mutation in cancer isn't random at all? What if it's actually following a hidden mathematical rule, one that could predict its behavior with, frankly, stunning accuracy? That's the mind-bending idea we're going to unpack right now.
Right. So we're taught to see cancer as biological noise. It's like a storm of uncontrolled, random mutations just wreaking havoc. But the author of this paper, Jean-Claude Perez, he looked at all that noise and asked a totally different question. Is there a signal in here? Is there some kind of hidden order that we've all been missing?
Now, before you write this off as some kind of science fiction, you should know this work got the attention of some very serious scientists. Professor Francois Gros, we're talking about the co-discoverer of messenger RNA here, he called the findings remarkable and of great importance. He saw that it could lead to a brand new way of classifying tumors. So what on earth did the research actually find?
Well, the entire theory, the whole thing, it all hinges on one beautifully elegant idea. The concept that our entire genome is trying to reach a single golden number, an ideal state of, believe it or not, mathematical perfection.
It's called the Human Genome Optimum, or HGO for short. The easiest way to think about it is like the factory setting for our DNA. It's just a number calculated from the ratio of its chemical building blocks, all the C and G bases divided by all the T and A bases. The paper's big idea is that there's one perfect optimum value for this ratio.
And here's where things get really wild. This number isn't just some random biological measurement. It's derived directly from a universal constant that you might have heard of, phi, also known as the golden ratio. The number that pops up everywhere in nature, from seashells to spiral galaxies. This simple formula connects the very blueprint of life to a fundamental number woven into the fabric of the universe itself.
And this is it. This is the number. 0.6909830056. According to this theory, this specific value is the perfect balance point, the mathematical sweet spot that the human genome is constantly aiming for.
So, is there any real-world evidence for this? Well, check out this chart. It's pretty fascinating. It compares the calculated HGO for 3 different genomes: a Neanderthal, an early human genome sequence from 2003, and then the much more refined one from 2013. See that yellow line? That's our ideal HGO number. And look at what the bars are doing. With each step in evolution and our own improving accuracy, the genome's value gets closer and closer to that ideal line. It's like our genome is on a journey, constantly fine-tuning itself toward this mathematical optimum.
Okay, so that's the big picture for the whole genome. But the research gets even more predictive when you apply the exact same logic, not just to the whole thing, but to each of our 24 individual chromosomes.
To get this next part, let's use a super simple analogy. I want you to picture a seesaw. That golden number, the ideal HGO we just talked about, that's the fulcrum. It's the perfect balance point right in the dead center.
So what happens when you calculate the HGO ratio for each chromosome individually? Well, it turns out none of them land exactly on that perfect balance point. Some of them, like chromosomes 4 and 13, have a ratio that's below the ideal. The paper calls these upstream. Then you have others, like 17 and 19, that have a ratio above the ideal, and those are called downstream. So basically, every single one of our chromosomes is sitting on one side of the seesaw or the other.
And these charts here, they show you that seesaw concept perfectly. The chromosomes are all ranked, from the furthest upstream all the way to the furthest downstream, creating this complete spectrum. On the left, you see those upstream chromosomes, and on the right, the downstream ones, and they're all positioned relative to that central balance point.
Okay, so we've got the ideal number and we've got this ranking of chromosomes on a seesaw. How in the world does this connect to cancer? Well, this brings us to the absolute core claim of the paper, a proposed universal law for cancer mutations. So here's the first half of the law. It's focused on a common thing in cancer called loss of heterozygosity, which is really just a fancy way of saying a chunk of a gene gets deleted. The law states that when this kind of deletion happens on an upstream chromosome, one of the ones below the ideal number, it causes a degradation of the entire genome's HLO.
In other words, it pushes the whole system further away from that perfect balance.
And now for the part that's going to bend your brain a little. When a deletion happens on a downstream chromosome, one of the ones above the ideal number, the law predicts the exact opposite. The mutation actually improves the genome's overall score, pushing it closer to the ideal HGO. Yeah, you heard that right. It's a shocking idea, a cancerous mutation, that from a purely mathematical point of view is optimizing the genome.
A claim that bold needs some serious evidence to back it up. So, how was this law actually put to the test? Well, the researcher went and analyzed data from a bunch of previously published scientific studies on cancer genetics.
The test set wasn't small. We're talking 240 specific tumor mutations, analyzed across 5 different kinds of cancer, including breast, prostate, and brain cancer. And these mutations happened on all sorts of different chromosomes, some upstream, some downstream, which made it the perfect data set to test this law.
So, what was the verdict? It's pretty staggering. Out of those 240 cancer mutations, the law's prediction was right in 227 of them. That is a success rate of almost 95%. The law correctly predicted whether the whole genome score would move away from or towards that ideal number in nearly every single case.
And this graph right here is just a stunning visualization of all 240 of those tests. That flat blue line, that's the HEO of a healthy genome. The jagged red line is the HEO after each one of those 240 cancerous deletions. The first 82 cases are all on upstream chromosomes, and what do you see? The red line consistently jumps above the blue line, degrading the optimum, just like the law said it would. The rest are on downstream chromosomes, and sure enough, the red line consistently jumps below it, improving the optimum. You can see the law holding up clear as day right across the board.
So, okay, why does any of this matter? What are the real-world implications of this? Well, if this mathematical order is real, it could fundamentally change how we understand and maybe even how we treat cancer.
Well, the paper suggests this could open the door to a totally new way of classifying tumors, not just by where they are in the body, but by their mathematical signature. It might even give us a way to predict how dangerous a tumor is, its pathogenicity, just based on where its mutations fall on that chromosome spectrum. It suggests that underneath all the biological chaos, there's a mathematical logic that we might finally be able to read.
Which leaves us with this one profound final thought. This single paper is making an extraordinary claim that a simple ratio, one that's connected to the ancient golden ratio, seems to govern the behavior of cancer. It's a radical bridge between the clean, pure world of mathematics and the messy reality of disease. Could a single number, rooted in ancient math, really be a key to decoding cancer's chaos? That's definitely something to think about.